Very excited to be here. Super interesting discussions this morning. I'm really happy to read in the instructions that it's about new ideas and have a lot of unsolved questions. So any feedback, welcome. And also you just see the highly dangerous metaphors we heard from Leo this morning. So I use this to communicate ideas, but the math doesn't depend on metaphor, right? I want you to be very clear about that. I think the mouse is delayed, so I'm going to use a laser. Very briefly, just to remind you of the formalism and notation. So everybody knows we have gene regulatory networks that constrain how individual genes can change their expression value. And that defines the gene expression profiles. For us, it's a state vector. And we don't care if it's RNA or protein at this point, right? A gene is active to some extent, that's the small x. And that are the gene expression profiles that we measure. And they change over time in a manner that is constrained by the network you see. And we can model it with ODEs and these functions that we've seen this morning. F, the large F here, which is essentially a vector field. And so my goal was always to see whether there's sort of a landscape there in some way. And I will tell you why that is useful as a metaphor. And these states define cell states because we talk about cell states. So it's just a vector, right? And they map somewhere to this landscape, and that trajectory is that this green curve that goes down. And so that's what we call the global quasi-potential landscape. Important global. Many years ago, Waddington proposed that, and he actually knew much more, as you can read in his posthumous book, up here in 1973. So these are not state space dimensions like X and Y on the right, but essential parameter. And even more abstraction, I will use this as a cartoon, a one-dimensional landscape where X is a state space. And then you have some sort of elevation, because when you have a landscape, you have elevation, and you need to know what it is. Very old idea, started with Max Delbruck. People have thought that these attractor states, the stable states with respect to all directions, represent cell types. And I think that's it's a good idea, the modifications of that. And many people have suggested that relatively independently of each other. Okay, the theory very quickly, also as a refresher. The two questions, right? When we talk about landscape, a little bit more as a metaphor, what we mean is some sort of energy, a scalar function on top of every state, X. And the other is how do we go from one state to another? And that's what I mean by attracted transition. It's not just go from a spot to another in the RNA velocity, but it's really going from one attractor to another, because that's a differentiation. What happens when you do so? So quickly, and that's these two things that are published. There's a noise-driven transition. If you have this Longchamp equation, then you can see you have stochastic fluctuation, and sometimes you hop to the other states. And then for many years, actually 30 years, people who knew about attractor state were debating how do you go from one attractor to another. And it's not at all clear. Many ways of thinking about that, and I will come back to that later. But the noise-driven state transition would fit into the theory that allows us to somehow come up with an idea of what is the elevation of this landscape. And many people here have also worked on that, like Jin-Wan and Jean-Hua probably will talk about that. For us, the easiest way is to think of it in the following way. So you don't have a gradient function that is your energy, because in this high-dimensional system, you don't have that typically. So this vector field can be decomposed into two, and there are many, many ways to do that. If you do it in a perpendicular manner, and you can arrive at the Frieden-Wenzel least action, we think that's the best way to characterize the quasi-potential energy, because it essentially penalizes for stochastic fluctuations to act against the deterministic function imposed by regular interactions, right? And I think Leo talked about that we have pure noise that can go in any direction, but there's a least action in that, it is a preferred direction at the end. And one can then show that one can formulate this Arrhenius-like equation in which the relative potential difference is related to the probability of going from A to the top, not to the next one, but to the top. And that's a totally damped system, so you don't get the energy back if you go down. It's only propagating to the top. But that constrains your system. It gives you the ability to have an estimate of how hard it is to act against the F, so the gene regulatory network constraints. And that will be useful. It turns out that, and I will show you later, this gradient part of this vector decomposition is, and we don't know what is G, is much larger than G for a subclass of networks. And that's important. That makes it possible for you to view the whole thing as a gradient-like system. I mentioned about attractor transitions. I think most people have settled down on this question that it is most favorable and convenient to view attractor transition as a bifurcation, because otherwise there are many other difficulties. So you will get to that, or some people call it a critical state transition. Both give you a directionality. That's very important for the later part of the talk. Even a bifurcation gives you an inherent directionality of going from one attractor to another. This is essentially, this here, right? So this is a directionality based on the sign of this delta U. Even if it's reversible, one direction will be preferred, and then we define the directionality of the transition between two attractors. Now, how do we know that state transitions occur via bifurcations? So for example, as most of you are familiar with these fold bifurcations, most textbooks will show you this diagram, bifurcation diagram. So going forward is mu, a bifurcation parameter, and you have the stable states, and it goes to an unstable state here. So also many textbooks draw this type of landscape. Now, it's a metaphor, I agree, but it's important because elevation plays a role, and the shape actually plays a role, because now what we have with single-cell analysis, we have entire populations, so we are not interested in the time evolution of a state vector, but the time evolution of your entire population, which is a matrix that you see here, and it has patterns, it's a population structure that we can exploit. So in one model, we assume that this is cloud of cells, and I agree, it is random noise that destroy all the deterministic drive, but they're still biased, and the way we model it is you can write it, so you don't know the function f, but you write it as a function of the eigenvalues of the Jacobian evaluated always at this attractor state when your bifurcation parameter changes. And this is just writing down as that to see how population structure changes. And then you can you add now some Gaussian noise that doesn't need to be Gaussian. You can then show that when the largest eigenvalue goes towards 1, which is a case of a bifurcation in these discrete systems, then the correlation between the genes reaches a maximum. And that can be used because we can measure this matrix here. So the goal is all about bringing it to a form that you can measure in single-cell analysis. So what we have is this index I, and many people have done similar things, that as you approach the bifurcation, the correlations between the genes would increase. That's not so counterintuitive, but you can appreciate now the attractor flattens. And so the cells are in a lower dimensional space because they go to these reaction coordinates that people have mentioned this morning. And the genes are aligned, they're more correlated, but the cells are more diverse. Some go already over the hump of the hill, others not. Therefore, the correlation between the cells decreases. And so therefore, the index I increases. And this you can measure. And we and others have published many papers on that. So I won't show you a lot of data. But there's another thing I'd like to tell you, and that is the consequences of thinking in terms of network architectures and landscape structure. So we think that given a network and genes, the landscape topography is fully determined in the deterministic system, right? And that's why you can predict all potential attractors and fates. You change the topology of the network, so the F, typically you don't change much, small quantitative changes, occasionally, which are bigger changes, you change the landscape qualitatively, okay? So on the top part, this is what is referred to as structural stability, where things don't change qualitatively, but occasional mutations, like homeotic mutations, would make an attractor disappear or a new one appear. And you're all familiar with those phenomena. We think that's corresponding to a qualitative change or a bifurcation due to a larger change in the this function F, and that is an innovation in nature. So what can we measure? We can measure state transitions and probabilities, and therefore predict what's the directionality of transitions between two attractors. And we can measure this index I to show how close you are to a imminent bifurcation. So the way we measure, we've just heard a few nice talks in the lightning talks. It's a barcoding, but it's a different modification here. So we barcode every cell, then the T0, they have to be expanded, and then you monitor the entire cell population. And the question is, from which given initial state is a cell state as measured by transcriptome derived at T0? That's what you want to know. And because of the heterogeneity of states, of microstates, it might be totally different, right? So now there's a trick here because you also need the statistics. You cannot deal with individual cells. So if you want to measure RNA-seq here and here, you get this nominal transition, but you need some statistics. And the way you do that is you minimally expand after transduction so that you get a population of cells that have the same barcode and very, very closely similar transcriptome. So it's a minimal diffusion in this transcription space that we call a family. And then you can split it, so you introduce a binomial noise, because one part of this family will be sequenced and represent the T1, and the other part will be then sequenced later on, which is then T2 here. And that's how we do it. And you can see that we can then label every cell according to the barcode. So the two levels you need to be aware of: your cell state, I, which is a position in this UMAP or transcriptome, and then cell lineage, J, which is where they came from, and that's the barcode. So it gets complicated, but I try to illustrate that with this animation. So here, you have cell state I, you go from gray to green, and then you also have the barcode J, this is barcode 1, it goes from blue to red. So you can see here the 2 time points. These cells here didn't change much between the 2 time points. Another barcode, like BC2, labeled these here cells at time point 1 and later spread all over the place. So this state attractor state probably diverges much more, whereas this attractor state is much deeper, cells don't change much. So you can do that. And then do this for many things. Not every barcode will be informative. Some are, some are not. And together with these are people involved, Joseph and Erin, Angelini and Hong Jiang, we started to collect data for that. It's very simple. It's just a counting of barcodes and cell types. You get, this is just a transition matrix. You get this matrix that predicts how number of cells change. And then you get this matrix exponential. And most things you can derive just by counting barcodes and cell types. There's a lot of problems here, right? You have to catch it before things go back and all these things. So you have to optimize everything. You have to expand a barcode into a family in which you know the barcode label.
This model totally deterministic.
Pardon?
This model totally deterministic.
This was deterministic, but there's a stochastic version in the making. Yeah, this is now, you just count.
Right, so it's totally deterministic at this point.
In this case, yes. But Erin's work, she has a stochastic model. This is just a result. I don't have time. You can actually then compute rate for transitions in both directions, from which you then deduce the net directionality. So, and this other measurement is index I skipped. This is published. Essentially, you measure, for example, in this case, we heard about all this target therapy. If you go from one state to another, this is now induced perturbation. Cells often go to this neural crest differentiation state. And if you color them according to this index, you can see a intermediate state, which has very high value. And this is another example, but just to show, to convince you that cell-cell interactions is a correlation, and by n matrices decreases and increase again, but gene-gene correlation increases. Here you can see, they can also be anti-correlated, doesn't matter. And so you see a peak, which corresponds to these intermediate states, yeah.
How do you define the criticality index?
Oh, that was the way, the previous slide. It's a cell-cell correlation and gene-gene correlation, just average. So you want to see that when cells are here, it's obvious that cells are more diverse, but less obvious that the genes are less diverse, they're correlated. It's probably lower dimensional space, but it mentioned reaction coordinates, so they go through this funnel or whatever, and they go then on the other side.
Can I ask you a quick question on the barcode stuff? I think you mentioned that some are more useful than others.
Yes.
What do you mean by the ones which are not useful?
These are the ones that happen to mark states that are highly unstable. And so in T2, they're all over the place.
But how do you know that that is the case and not something else?
Well, they're just,
like that it is possible that it's not about dynamical system and somehow some barcodes just happen to traverse all the spaces.
They have to. I don't know what you mean. So some barcodes
which are tightly constrained in the high dimensional space,
yes,
and some which are not.
Yes.
Do you have a causal proof of that? It was like, how do you know that? Like, how do you know what led for them to be dispersed?
We assume they were in a state that dispersed much faster than not. You know, the single-cell sequencing turned out to be extremely powerful. You know, you have UMI, you have barcode, we control for everything, barcode hopping and sequencing error. I think it's super robust. I don't think there's a technical error there. But
are you suggesting that it could be random?
It could be random as well, yeah.
Could be random.
Or it could be, I mean, there could be a technical reason of the barcode assignment in single cell is very non-trivial. So you can easily, even a couple of SNPs, you may call them different barcodes.
No, I agree. We check for that. I had a very beautiful slides. We do all one Hamming distance neighbors, and so they don't cross over. We study the barcode hopping phenomenon.
My ding didn't work, but you have under 3 minutes.
You can
okay. So this is the coolest slide. Okay, good. Then I'll skip the data because there's no data for that. So a big idea then is, and that's what I need help from everybody. You have all these clusters. Can we then, if we could compute some energy to that, the many approaches, AI-based, entropy, whatever, I don't quite understand them, but the goal would be, are they ordered in some gradient fashion? Then you could have transition rates between attractor states, and you want to see what the nature tries to avoid these non-transitive sets. Because if that would be the case, then there wouldn't be development. And the reason why we have a gradient-like system is why all these other fascinating approaches, people doing just landscape topology or geometry to explain evolution and development, and also cancer, and I will show you here. So my dream is to map everything into a linear ordering, and given a directed graph, that's possible. It's not unique, but at least using some transform, you can show from the transition matrix that you don't have much non-transitive sets in your transition graph between all the attractor states. And then it will be a gradient system, right? So just the energy will tell you which state can go into which other. So you can achieve a partial, which means non-unique topological ordering of your attractor states. Now I need to introduce you another concept many people are not familiar with. Yeah.
Surely the ordering would change based on the signals you put in, right? Even the example we just heard of AT.
Yeah, I know, but this is
So I'm just wondering what you mean by unique, because
no, but
it
no.
seems environment dependent.
Even a directed graph, the ordering is non-unique because
But I don't understand if the graph depends on the signals that are present or the environment or
no, this is spontaneous. It's spontaneous, right? I'll show you. I'm going to discuss, but
okay.
I'll show you just this. And all the network I talk to you, there's a space of all possible networks that can be mapped into a space of all possible network topologies. And people have been doing this for years. It's just not so well known. People characterize a chaotic and an order dynamics type of network based on the dynamics. And then life is at the critical transition. And so we simulated that and look for whether critical networks that we think most living systems are they avoiding non-transitive sets. That's a new idea. And at least in simulations of network, it really looks like critical networks have much lower propensity to generate non-transitive sets in the attractor transition graph. And then we'll show that in cancer, this is destroyed due to mutations, and you get these non-transitive sets, and cancer cells are trapped into these circles. So I don't have time to show you the data, but that's where we are at.
So what we can take a question while you set up. Is that okay? Do you mind if he sets up while we have maybe one or two more questions for Sui? Anybody else? Maybe my question, if you're not, it's okay. You can unplug if you want. I can just ask you.
Fine, yeah.
Yeah, you can plug it in.
Let's see.
Let's see. Yeah. There's a question here.
Oh,
Okay, thank you. Sorry, I
Okay. So,
really
Sui,
thought- what was your name?
Jim.
Huh?
Jim Wang.
Jin.
Yeah, sure.
Yeah. Why do you insist on saying that the gradient force is dominating?
Oh, that's initially our assumption.
Yeah.
It only works if it does. Otherwise, but then we have data to show, simulated data of simulated networks. There's one support.
Which data? Which data are you?
I was supposed to show them later on, but
No, I mean just the specific cancer data.
yeah, leukemia, because there you can have multiple time points and then your mutations. But also, otherwise, the gradient. People. I don't know. I think it's Eric Seeger here. But the fact that people can just look at the geometry to explain development is really suggests that we have a gradient-like system. And the question is, why do we have that? And that's it.